97. Continue. By mapping UTOs, we could report our results at the species level. This figure shows distribution of most abundant genera. As you see here, Streptococci, Granulicatella, Rootzia, and Actinomyces dominated most samples. There were low amounts of strict anaerobic bacteria, mostly ranging far below 1% of total reads, or missing completely. These figures illustrate alpha and beta diversities. Most samples had species richness between 30 and 79. Figure C shows unweighted qualitative unifrac analysis, and D show weighted quantitative unifrac analysis. And we had one extreme outlier, sample 28. This was a sample totally dominated by E. coli and was collected from a patient suffering from severe constipation. We found the microbiota 25 centimeters from the ileocecal valve to be oral-like and strikingly different from the colonic. Most frequently detected bacteria were Streptococcus, Granulicatella, Actinomyces, Solobacterium, Rootzia, Gemella, and TM7. Our results are in sharp contrast to most contemporary authors and medical textbooks. Some claim our population is too small to draw this conclusion. So, our second article in Clinical Microbiology and Infection, we expand our cohort to 150 patients and report exclusively cultivable results. This cohort is large regarding surgical sampling from the small intestine. By culturing these samples, we can tell which microorganisms that are real inhabitants in the lumen. The microbes reported are viable and not only dead remnants or DNA related to food. By cultivating samples from 150 patients, the conclusion persists. Bacteria related to the oral flora dominate. Somewhat surprising, most samples harbored a much scantier flora than previously reported. Contemporary authors claims 10 to the 8th colony forming units per milliliter. We find 10 to the 4th or less in 79% of the samples. Colon-related bacteria, thought to be Enterobacteriales, strict anaerobes, and Enterococci, were only recovered from 14% of the samples. Oral-related bacteria was far more frequent. In addition, 56% grew Candida, mostly albicans. And as you know, fungi is consequently missing in 16S deep sequencing studies. Interestingly also, Saccharomyces did not grow at all. For me, this questions the relevance of deep sequencing results indicating Saccharomyces to be a relevant actual colonizer in the human gastrointestinal tract. Achtung et al. From 2018 referred in this article, support this. Achtung et al. Showed that a complete bakery yeast-free diet eliminated reads from Saccharomyces in stool samples. This is the results concerning fungi. Saccharomyces do probably not survive the acid and digestive enzymes. However, another diet-related fungi, Candida kefir, probably do. We also cultured 10 different lactobacillus species, some of them related to food. And if you are interested, they are all listed in supplementary material. Our third article is in progress. We have 120 Yale samples from 60 obese patients. Most samples are extremely weak. PCR step were now run for 45 cycles, and we included 20 negative controls this time. 10 negative and 10 positive negative controls, spiked samples. By doing this, a median of 75% of the reads were removed as contaminants. One sample had 98% of the reads removed as contaminants. Only a couple of Corynebacterium species were left. We hope to present this material ahead of next summer. Through careful review in databases, I did find 3 studies based on clean surgical sampling directly from the distal ileal lumen. Only in the 50s and 70s, researchers were allowed to puncture the small intestine when they did elective surgery on other organs. ReC would never approve these studies today, but we forget about them. This is important work and should be addressed in reviews and medical textbooks. I believe it's unfortunate that these studies seem to be skipped in favor of case studies on mice. Cragen et al. From 1953 did a study on 14 young, healthy women undergoing elective gynecological surgery. They found the terminal ileum had sparse or even no growth. Bacteria were chiefly those gram-positive species that are usually found in the mouth and throat. Kurodi et al. Did a study in 1978 on 8 obese patients undergoing gastric bypass. They found the terminal ileum harbored a relative sparse flora, contrasting with a large concentration of microorganisms on the other side of the ileocecal valve. Bentley et al. Did in 1973 a study on 10 patients undergoing elective cholecystectomy. The terminal ileum generally had lower colony counts with a higher ratio of aerobic to anaerobic bacteria. So my conclusion is that the small intestine, through its whole length, contains a sparse flora, mostly related to the mouth. Because of a difference of 10 to the 8th in microbial density gradient, there is a considerable risk of contaminating ileal samples collected by transcolonic procedures. And I do not believe fecal material is a relevant representative for the overall gut microbiota. Finally, I have some questions regarding research in host microbe relationship studies. Researchers working within the microbiota field needs to have some clear thoughts about the assumed pathophysiology. If you are working with fecal samples, what do we know about the colonic intestinal barrier function? How do microbiota affect intestinal permeability in colon? We need to know if and how passage of small molecules occurs between cells, and more about trans-epithelial pathway in colon. Another question we need to ask is how relevant mouse models are for human physiology. Mice are rodents. They eat exclusively plant food. And they are coprophages. They eat their excrements. There should be no surprise that microbiota studies from the small intestine in mice are similar to the colonic. Mice is considerably different from human omnivores. We eat food of both plant and animal origin, and we do not eat our own stool. Rats could probably be more interesting to study. Rats have more similar gastrointestinal tract with longer small intestines and do also eat food of both plant and animal origin. Metagenomic data should probably include both 16S and ITS primers to get a complete picture of the microbial ecosystem. Contaminants and food remnants could also be a considerable bias in culture-independent studies. Is it necessary to standardize diets before comparing cohorts? And in our search for host microbe interactions, could mouth and saliva studies be of greater interest? The average person swallow more than 1 liter saliva per day. In my opinion, we need to discuss this and be aware of these issues. So thank you for listening.
Thank you, Heidi, for a very interesting talk and also thought-provocative. I think it's really interesting studies, and you question a lot of literature, and you also question a lot of the work that other groups, including ours, are doing. Mm-hmm. So first question from my part is, since you find that it's an oral-like microbiome in the small intestine, would sampling from the mouth be most convenient? And how do you see this? How should this data be described? Should you, if you believe that the gut microbiome is playing a role, should you report data both from the mouth and the fecal microbiome? Because I think you need to have some accessibility to samples. So do you have any comment to that?
Probably the best would be to sample from both places. The Ibsen study focusing on morbus Crohn and ulcerous colitis we have, for the last sampling year, included oral studies, oral samples in addition to the fecal sampling.
Yeah. And the Saccharomyces you found, do you think that's from the diet?
No, we did not find any Saccharomyces, but we know that Saccharomyces grow very well on our plates. So,
Yeah.
so if there were viable Saccharomyces in the small intestine, it would have shown on our plates. I believe so.
And my final question is, you present data from a bladder cancer cohort. Do you think that's representative of the healthy microbiome of the small intestine?
That's a recent question. If you read my latest article, you will see that the oldest people in our population had more Candida albicans than the younger. I think age will play an important role. But I believe also that the older studies presented here, collecting from the terminal ileum, even closer to the ileocecal valve, do support our findings.
Yeah. Interesting. Any other questions or comments from the audience? Hanne, did you're also welcome to pass questions on the chat, and Hanne will read it and follow it.
I have a comment. Hi.
Hi.
Hi. Very nice talk. First of all, the first comment, yes, regarding the quality of the fecal microbiome analysis, I totally agree with you, especially if we consider that the mucosal counterpart of the microbiome probably is what has a major role in interacting with the host. And the second comment, in line to what Marius commented regarding the second paper, I mean, there are some articles and some study that describe that in some pathological conditions, such as, for example, also the liver cirrhosis, there is a migration from oral to the second part of the intestine, indicating that potential is part of a pathological condition. So if linking to the Marius question, do you have also in healthy patient some data regarding this oral counterpart of the microbiome into the ileum?
I'm not sure if I got the question right, but the youngest.
You have the data also in healthy patient,
no,
not only with biliary cancer.
No. Also, ReC would never allowed me to do this study on healthy patients. This is the best we got. Additionally to the older studies that I referred, that I believe is important work. But the youngest people in this material is the people with less growth. And we also, if you read the second article, we find a slight association between constipation and more growth of colonic microbes. So I believe that it is the deceased patients that had a small intestinal microbiota, perhaps in a colon-like way. The healthy one is even have more scanty flora than this work presents.
Heidi, another question. I mean, I think this is very interesting, but the question is how to use it. And when we talk about microbiome, we often talk about, metabolites produced by the microbiome. I've been talking a lot about TMAO. Others are talking about short chain fatty acids and so on. Do you think that mouth samples or saliva could be more suitable materials for gathering and measuring such metabolites or other metabolites?
If you're talking about small fatty acids,
it was just an example. I mean, we are still mapping this, right? But could saliva be an additional sample to look into?
Yes, also this is perhaps a little bit provocative, but I just want us to be aware of these issues. We need to be aware of them as researchers, and I believe mouth and saliva studies should be considered in some questions more than feces. The small chain fatty acids is important for the health of our colon, because the small chain fatty acids are energy substrates from the colonocytes. So I do not believe that small chain fatty acids are important in the small intestine, but as I pointed out here at the beginning, I'm not sure how much do we really know about the transepithelial pathway in colon. It possibly more. It is possible that colon is more penetrable than we recognize today, but we should be interested in the colonic intestinal barrier function, and a lot of not standard medically educated treaters today are talking about gut leakage. You don't talk about that much at hospital, at the gastro medical department, I believe, but it is perhaps a more important question. Perhaps many of our inflammatory diseases have something to do with our guts being leakage, and it could be that microbiota is that microbiota really affect the intestinal barrier function. If you have the wrong microbes in your colon, perhaps there will be more leakage also from colon. I don't know these questions. I just done some research on the small intestine that I believe should be considered for everyone working within this field, because we need clear thoughts about the assumed pathophysiology that we believe in.
Yeah, thank you. Are there any neurologists here today? I mean, we have some collaboration with the neurologists at Oslo University Hospital, where we are sampling from different sites, fecal samples, but also I think it's either oral or saliva samples, and then also biopsies from the carotic plaques in patients. So that was also an idea to see how this microbial signal could vary at different locations. I do not remember the details on that project. So, but if nobody is here, that's maybe something we could discuss at another occasion. Any other comments from any other in the audience? If not, I want to thank Heidi for a very interesting talk. And Hanne, can you just tell us when the next meeting is?
So we will not have a meeting in November because we have our conference on November 16. We're very excited for what we're planning, and we're still accepting abstracts. So please send them in to us if you have them. And some more information on the conference will come very soon. We're making some great plans, and I think we'll have a good conference to look forward to.
You can say that we are trying to set up a TV studio-like setting. I mean, it will be boring to look at Zoom from laptops all day, so we'll probably try to have a studio. And then it will depend on you guys what we are talking about. You know, it's the researchers who will drive the agenda. We have one invited speaker, but apart from that, we will try to see what pops up from abstracts. If there are not so many data this year because of COVID, you could also come with some suggestions for discussions and so on. I mean, we could also try to use this format to test out other ideas or having conference. So I think we should just take the opportunity to test out things and see how it works. I mean, this year we are forced to meet digitally, but it could also be if we make good use of new platforms that we could maybe have some more frequent meetings also this way. Yeah. So I think it will be. I hope as many as possible will meet up at the conference, and I think it will be free of charge, right, Hanna?
Free of charge this year, and
yeah,
easy to access. And
but we still want you to register because we also have to report the network activities and so on. But I'm very eager to test out some new platforms. I think this is a good opportunity to do that.
Mm. So any suggestions, please email us at the post@microbiota or directly to me or Marius or Johannes. We'll be happy to hear.
Send it to Hanna. She's good at these things. Yeah. Okay, it was nice seeing some of the faces in the Zoom window. I see you there, Dag Henrik. Hello. Thank you again to Hanna, to Heidi. And,
thank you, Heidi. It's a very good presentation.
In November. Yeah.
Yeah.
Bye.
Okay. Thanks, everyone. Bye.